Biogenesis of microRNAs. MicroRNAs, they are small, non-coding, double-stranded RNAs. Approximately, their size is between 19 to 22 nucleotides. miRNA originates with single-stranded RNA molecules that forms a hairpin secondary structure. MicroRNA originates from the genomes and they are capped and polyadenylated full-length precursors, means their genes are present on the genomes and they originate in the form of pre-miRNA. How is the biogenesis of miRNA or how miRNA are formed? There are primary miRNA transcribed in the nucleus and their genes are present on the chromosomes. Once they are transcribed in the nucleus, then the size of those transcribed primary miRNAs is in several kilobases that have 5 cap end and 3 poly A tail. Once they are cleaved in the nucleus by drosha cleaves it, that is an enzyme, into 70 nucleotide hairpin transcript. So once they are transcribed from the genes, then the next step is drosha enzyme cleave it or cut it into 70 nucleotides. It is called as pre-miRNA. Now this pre-miRNA is transported to the cytoplasm by a protein. The name of the protein is exportin 5. This protein exports the mi or pre-miRNA through the nuclear pores into the cytoplasm. Once the pre-miRNA reaches in the cytoplasm, it is cleaved or cut by dicer enzymes. Dicer enzymes, they are RNase 3 enzymes. It cut it into 19 to 22 double-stranded transcripts, that is the mature miRNA and ready to function. Here is the diagrammatic view that how is the biogenesis of microRNA. Here is the genes, this is the DNA, and the first step is the transcribed in the nucleus. In the nucleus they are transcribed. Here is the transcription. Then these large fragments, they are cleaved in by the drosha enzyme. This is the drosha enzyme. Once the drosha enzyme cuts it, then the third step is transported to cytoplasm by exportin. So now here, this is the pre-miRNA. This pre-miRNA through the nuclear pore is transferred to the cytoplasm. Once they reach in the cytoplasm, here is the cytoplasm, then they are cleaved by dicer enzyme into 19 to 22 base pair or nucleotides. So this is the pictorial view that how is the biogenesis of the microRNA, their genes on the genome. Then they are transcribed, cleaved by the drosha, and pre-miRNA are produced. Then pre-miRNA transported into the cytoplasm. Here they are again cleaved with the help of dicer enzyme, and the mature miRNA are produced between 19 to 22 base pair. Another illustration of the microRNA processing. This is the pre-miRNA. Drosha cuts it into pre-miRNA. This pre-miRNA, with the help of exportin 5 protein, moves into the cytoplasm. Here is the dicer, and dicer converts it into the mature miRNA. This is another view of that how biogenesis of microRNA processes. This is the nucleus. Here, this is nucleus. Here is the cytoplasm. What happens in the nucleus? Pre-miRNA, then drosha. Drosha converts it into pre-miRNA that can be approximately 70 to 72 nucleotides long. Then it is with the help of exportin 5, it is exported into the cytoplasm. This is cytoplasm. Within the cytoplasm, this pre-miRNA, with the help of dicer cuts it from here. With the help of dicer, it is converted into mRNA duplicates and the mature mRNA is ready to function. So this is that how biogenesis of mRNA takes place in the nucleus and in the cytoplasm. So what is microRNA? Small non-coding double-stranded RNA molecules, and their size is somewhere between 19 to 22 nucleotides long.